Hi, good night, everyone. As Chris said, I'm Stefanie, and I'm going to be talking to you today about my research along with your very own Amy Deacon, looking at social behavior in caterpillars, and specifically looking at how these caterpillars that you're seeing on your screen right now, how they coordinate their group behavior. Are you guys seeing my bar on the side here with everyone's picture? I'll just minimize this. Okay, right. So yeah, as I said, this work that I've been doing with Amy, looking at social behavior in caterpillars, and I think it's a really nice project to share with all of you guys because it really all started from a chance observation of a group of caterpillars doing a kind of unusual and interesting group behavior, and we were able to scale it up into quite an involved study looking at a whole range of aspects of these caterpillars' communication mechanisms and sensory mechanisms. So I think it's really in keeping with, I guess, the spirit of the club and the goal of the club to promote natural history. And it also highlights the important role that natural history observations still have in directing biological research, and especially in a place like Trinidad, where we still have so much to learn about our own biodiversity and animal behavior. So yeah, thanks for being here, and I hope you learned something, and thanks for inviting me to speak. Okay, so how I want to do this is I'm going to start off very briefly giving you some background information into the kinds of questions that we are interested in this field, just to kind of set the scene and get us all thinking along similar lines when we get into the actual observations. And then I'm going to go through what we observed and the research questions that came out of it. Then I'm going to talk a bit about how those observations informed our hypothesis development to explain the behaviors, and finally going into how we tested these hypotheses through experiments, and then looking at how all these findings fit together to give a pretty decent explanation of the behavior that we initially observed, and finally what we still don't know about these behaviors. Okay, so one of the, I guess, very general questions in sociobiology is, how do animals coordinate large-scale group behaviors? So thinking more in terms of the mechanisms that are going on inside organisms that allow them to perform group behaviors. So thinking less in terms of the adaptive reasons, but more in terms of less in terms of why and more in terms of how. And these are the kinds of behaviors that I'm thinking about. So very large collective motion in birds, cooperative foraging in ants or bees, and things like that. So behaviors that are way beyond the scale of each one of those individuals that are taking part in the behavior. So questions like, how do these animals communicate and interact to produce these complex group behaviors? Do different individuals have different roles in these behaviors? So do we have some individuals that are leading the group and others following, or is it something else? And perhaps most interestingly, how much do each of these individuals actually know about the overall pattern that they're producing? So one thing that we often find when studying all these different kinds of collective behaviors is that often natural selection stumbles upon the same kinds of solutions in very different systems. So everything from bacteria to humans, we see some of the same mechanisms at play that help us coordinate group behaviors. And the interesting thing about it is they often leave particular signatures that we can see across all these systems. And this actually helps us figure out what's going on in a given system. So more on that later. Okay, so these are the kinds of questions that were on my mind when I came across this group of caterpillars near my home in Lupino. So I came across a group of about 400 caterpillars on a guava tree. So like we can see in the picture on the far left. And they were performing a range of really interesting coordinated behaviors, resting together in a patch on the tree like this, also feeding all together, all pressed up together, like you can see in the other picture, and moving between different feeding and resting sites together as well. So this was a level of behavioral coordination that I personally wasn't really aware even went on in caterpillars. So the naturalist in me wanted to find out more. So I started observing them kind of over the course of several days to see what I could learn about about the behavioral pattern, any kind of hints I could get as to how it was controlled and what was going on. So after observing them over a few days, I noticed that they had a very distinct pattern of behavior over the diurnal period, so over the day, over the course of the day. And the first thing that I noticed is that they always spend the daylight hours from around 6 a.m. to 6 p.m. in the same resting patch that I identified in the other slide. So from 6 a.m. to 6 p.m., they always stayed completely dormant in a tight aggregation and really didn't move at all for about 12 hours of the day. Then around 6 p.m., when it was just about starting to get dark, they would start moving. So they would start off with occasional bursts of movement, eventually resulting in the whole colonies becoming like a swirling mass. And then one individual would start off walking up the tree. So let's say this individual here, they'd start walking up the tree, and directly behind them, another one would follow, and then another and another, until you had these really visually striking nose to tail processions of caterpillars going up all the way to the top of the tree. So they went up the top of the tree in these processions, and then when they got to the top of the tree, they would all aggregate again and feed, all pressed up together again on leaves. And then when it was just about starting to get light again, they would return in the processions back to their home patch, and they would fall dormant again, and the cycle would continue like that. So as I said, I was really intrigued by this, and I was interested in finding out how these caterpillars, which we generally don't consider to be particularly cognitively advanced or particularly socially interesting, how they were performing this level of behavioral coordination. Because it wasn't just a case of all of them kind of responding at a particular time and descending on mass to another part of the tree. They were doing this very deliberate seeming group behavior of going kind of one at a time all the way up. So I wanted to know, how is it that they decided when to get moving? How is it that they formed and maintained these aggregations and processions? And also, how did they navigate between sites? So I reached out to Matthew Koch, who is an entomologist and lepidopteran expert, who is also a friend of the club. And he told me that these caterpillars belong to a species called Hylesia metabus, which was a kind of saturnid moth. And he also let me know that the genus that they belong to, Hylesia, contained several more processionary species. So this wasn't the only one. So here's some pictures of some of the other processionary Hylesia species. So even though this behavior was well documented, it hadn't really been studied very much from a mechanistic point of view. People had kind of recorded that it was happening, but no one had really looked very carefully at how it was happening, at least in the case of Hellesia metabus. There was some work on one of the species, but generally, we still didn't really know how they were doing this. So I decided to continue looking at this colony on my tree and see if um, there were any hints as to what was going on if I kind of studied them long enough. And the lucky thing was that they were very tolerant of me. So I was there within a few feet of them, and they didn't really react to me at all. So I was able to really get a front row seat at the behavior, and I looked really carefully to see what was going on, and so I just started writing down everything I saw them do. I developed hypotheses on how they were doing it, and after a few days of watching the cycle play out, so this kind of daily cycle of resting, walking in procession, feeding, and coming back down, something different happened. I noticed that there was another group of caterpillars that had become very much intermingled within the first group. So if you take a look at this picture here, this black individual and most of these other guys in the back here, that was the initial group. And at the front, you can see some caterpillars looking a little bit different. So we can see one guy here, 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 and a few more interspersed. So I saw this new set of caterpillars enter the group, and they were doing all the same behaviors that the initial group was doing. They were resting in these daytime aggregations. They were even walking off in procession with them. So we can see, you may be able to tell here, these kind of less spiky ones were the new group of caterpillars. So they were walking in procession, they were feeding with them, they were doing everything the same. And to such an extent that I thought that maybe they had just gone up to a different instar. So maybe what it was that they had developed and there was some kind of polymorphism in this species where some individuals were big and hairier, and some were looked a little bit different. So I reached out to Matthew again to see what he had to say about this. And he actually let me know that this was actually a different Hylesia species that had become intermingled with this first group. And this one was called Hellesia Nanas. So this is when it got really interesting, because as far as we could determine, this was the first time anyone had seen, or at least anyone had recorded, two processionary species coming together and forming mixed species processions. So a first for Hylesia, or really any other processionary species. And it was interesting in a kind of more general sense because it was an unusual level of group coordination they showed an unusual level of coordination for mixed species groups. So here were two species that were actually not just coming together, but performing quite an intricate range of behaviors together. So usually when we see mixed species groups, the most you'd expect is that they would be maybe attracted to each other or attracted to the same location. But here we had caterpillars that were engaging in the same behavioral cycle, performing and walking in these processions, feeding together, and the works. So we thought this was really interesting because of that as well. So the first, I guess, obvious question that occurred to us is, what was the adaptive significance, if any, of this behavior? Was this some kind of new mutualism between two species? Was it even a parasitic relationship? Or was this just an artifact of a shared evolutionary history between the two species? But unfortunately, we couldn't really determine much about the adaptive significance from this one encounter, but we were really more interested in what it told us about the mechanism behind the behind the coordination. Because it did seem to suggest that these species actually shared a mechanism for their group behavior. Otherwise, why else would they be joined up together? And the other thing that it pointed out is that the mechanism was robust enough to preserve the group behavior. So even though you had two species coming together that didn't usually spend time together, the emergent behavior of the group, walking off in processions, and the whole daily cycle of behavior, that was preserved. So that in itself told us something interesting, that something interesting was going on in terms of the specifics of the mechanism behind the communication mechanisms that were going on in both species. So because of this, we kind of tweaked our research question from just looking at how do Hylesia metabus achieve behavioral coordination to how do both Hylesia metabus and Hylesia nanas achieve behavioral coordination, and what do these mechanisms tell us? How do these mechanisms explain why we were able to find this this unusual interspecies group behavior? Okay, so we went back to looking at the mechanism, and we were paying particular attention to the transition between resting and feeding activities. So we thought, well, if we look directly at how they're moving from this state of dormancy to processionary behavior, there might be some clues in there as to how they're doing it. And luckily, we were able to do this both before and after Hylesia Nanas entered the picture. So we had a really good side-by-side. And we found a couple of interesting things. So some of the key observations were that, as I mentioned before, the shift in behavior always coincided with the transition between day and night. So usually when it started to get a little bit darker, you would start to see a little bit of movement, and eventually they would all walk off in procession. So we felt that there was some kind of daily varying stimulus that was controlling the behavior in the general change in behavior. And we suspected that there was probably an effect of light, but it could have been something else that changed over the daytime period. So it could have been like a drop in temperature or something like that. We also found that when it did get dark and a few individuals started to move, they always seem to be cascades of movement. So when one individual moved, it seemed to stimulate the ones in the adjacent individuals to also move, which would then seem to cause the ones next to them to move until you had this whole group coming to life. Okay, so it seemed as though there was some kind of positive feedback response, where movement in some individuals caused a cascade of activities until they were all moving. Another thing we noticed is that they always took the same route up the tree. So because of this, we suspected that there was maybe some kind of pheromone trail that they were making use of, that perhaps when they come down to the patch, they deposit a pheromone trail, and then when they go back up the next night, they follow the same trail back up. And in addition to those, we actually use a pair of forceps to experimentally manipulate processions to see how individuals responded to changes in their local environment. And the first thing we tried was removing the individual at the front of the procession. So trying to figure out if the caterpillar at the front, if there was anything special about the caterpillar at the front. So did the caterpillar at the front know the way, or was it the most motivated individual or something like that? So what we found is, in most cases, when we removed that front individual, it didn't really change the behavior very much. So when we removed the one in front, in most cases, the one behind would just kind of assume the position of the one in front, and it would just continue on like that. So this suggested that there wasn't really anything inherently important about that individual in front. So it appeared as though there weren't any leaders in the group. It would just seem that any individual could lead a procession. And the second thing we tried was holding back sections of processions. So we used our tweezers again to just pin down one individual in the line, and recorded how the others around it responded. And what we found is when we held back one individual, the one directly in front usually stopped, and that would result in the one in front of that one stopping. When one lost contact, the one in front would stop, and then because that one would stop, the one in front of that would lose contact and stop, and you'd have this kind of domino effect all the way up to the top of the procession. So we suspected that the maintenance of the processions may have been down to these to stimuli from adjacent individuals. So, and we suspected that it might have been a tactile cue, but we do tactile in terms of touch, because we saw that when contact was lost, they would change their response. So we suspected tactile, but we weren't sure at that point yet. So when we took all of these things together, we found that they started to paint a picture of how this behavior might have been controlled, because all of these kind of behaviors, so stereotyped responses to local stimuli, positive feedback, and a lack of leaders, are all hallmarks of a particular kind of mechanism that we see in a lot of systems, which is called self-organization. So what is self-organization? So systems are considered self-organized when group behavior develops from many interactions between identical individuals, all responding in stereotyped ways to stimuli from adjacent individuals. So this just means that each individual is only aware of the stimuli in its immediate environment, and they have very set stereotyped ways that they respond to stimuli. So in this case, things like the tactile stimuli from the rear, we felt the loss of the stimuli at the rear stopped the caterpillar from moving forward. So things like that. So maybe a combination of, in this case, a combination of tactile and chemical cues might have been responsible for the behavior of each individual. And this was consistent with what other people have found in caterpillar social groups, because even though there are a few other caterpillar species that show these processionary behaviors and things like that, we haven't actually found any examples of behavioral differentiation leaders or long-range communication in Lepidoptera and larvae or caterpillars. So this just means that unlike things like bees and wasps, where you have different behavioral castes, so you have some workers and reproductive individuals, all different types of individuals in caterpillar groups, we're not really aware of any system that has that kind of differentiation. So as far as we know, in all caterpillars, all the individuals are basically the same. And again, there's no evidence of elaborate communication mechanisms. So this seemed like the kind of thing that would be going on in a species like this. So I was thinking that this might be kind of hard for people to visualize. So I thought I'd give an example of something similar in humans, so we can imagine the sensory environment that these caterpillars are experiencing. So I have a very contemporary reference of people in a party performing conga lines. So if we think about being an individual in a hall, let's say, with music going, and people start forming a conga line, you'd realize that no one individual is really responsible for that behavior. So you just hear music going and you follow a simple rule, right? So you feel someone put their hands on your shoulders, and that means that you have to move forward and put your hands on someone else's shoulders, and then they know that they have to go and put their hands on someone else's shoulders. So they're all following the same set of rules, but you get this overall emergent pattern of this huge line snaking around the hall or the party or whatever. So this is kind of what we thought might have been going on in these caterpillars. It was just these local rules leading to the formation of processions with no one individual really being aware of the overall behavioral pattern. And self-organization also seemed like a good explanation for why the second group of caterpillars, Hylesia Nanas, were able to integrate so seamlessly into the colony without affecting the overall behavior. So think about it like this. In our conga line example, if another group of people came in, let's say from one of these doors, there's a party in the other room. If one of those if another group came in and saw this, well, and there was this conga line forming, they would very easily be able to participate in the behavior if they had the same, if they were aware of the same rules, right? So they also know that you have this thing where someone puts their hands on your shoulders, you go to the next person, and they're able to perpetuate the behavior and still have the emergent behavior be preserved. Compared to something like the image on your right, which is a highly choreographed and an organized dance, where even if a second group of dancers came in with all the same rules, so the same mechanism, it would be very hard for them to kind of slot in and still preserve the overall dance, because it's way too complicated with different individuals playing different roles. So we felt that self-organization could explain why H. Nanas was able to get in there and do all the same things without confusing the overall pattern. So based on all of this, we hypothesized that a shared self-organized mechanism accounted for the interspecies gregariousness and processionary behavior in these two species. And we also felt that the mechanism could involve chemical and tactile signals as well as light. So how do we test something like this? So luckily for us, there's a kind of a set way to test possible examples of self-organization. So it's a kind of three-step process. The first thing was the in-situ observations and manipulation. So the bits of the story that I already explained, where we looked at the group behavior in situ in the wild system, and try to figure out what was some of the stimuli that might have been involved. So, and that's how we developed our hypotheses, which I've already outlined. And then we move over to performing experiments in the lab. So taking the behavior into a more controlled setting, removed from environmental stimuli, and seeing if they're able to still produce the pattern. So self-organization would predict that since the behavior arises just through the interactions of all these individuals without the need for external stimuli, you would expect the behavior, if it's self-organized, to still arise even in a featureless environment. And then we did some X-Situ group experiments, which also helped us to figure out some more about the rules and the specifics of the behavior. And then we scaled all the way down to experiments on individuals and pairs of individuals. So self-organization would predict that in a self-organized system, each individual will behave in exactly the same way as every other. And when you scale up all these individual behavioral responses, you should be able to account for the global properties of the system. Okay, so starting off with these X-Situ group experiments, we wanted to know if groups could form, so both groups of H. nanas, H. metabus, and a mixed species group, whether they could form aggregations and processions just through the interactions between individuals without any external stimuli. So we placed groups that was spaced, groups of about 20 individuals spaced equally apart in a featureless arena, and we just watched what happened. So this is a mixed species group. So as you can see, they very quickly start moving towards each other. Well, they start moving, and then when they get in touch, when they line up with another one, they kind of spontaneously aggregated and formed their processions. This is the H Nanas group, and something similar. So as they walk, they bump into each other, and then they stick together and they form their processions. And finally, in the H metabus group, something similar is going on here as well, right? So just by virtue of being in contact with each other, they're able to form these aggregations and processions. And this actually held even in groups of as small as 2 individuals. So we did something similar with just pairs of caterpillars. And again, we found that they would usually spontaneously aggregate and form processions. So this was highly consistent with a self-organized mechanism. So that's what I just said. So groups spontaneously aggregated and form processions without external stimuli. The other thing that we tried was to test the start and stop rule that we had identified in the wild colonies. So remember I said about that we held back sections of processions on the tree, and there was this characteristic stopping of each individual in front. So we wanted to see if this held in this environment in the absence of any external stimuli. And again, we found that usually when we held back an individual, the one in a procession, the one in front would stop. And we tried, we wanted to see as well if this was a result of tactile stimulation, because we already knew that there was a response, but we weren't 100% sure what they were responding to. So we held back sections of processions and we replaced it with a little brush. So we just brushed the rear end of the individual in front, and this would usually stimulate them to move off again. So this supported this idea that the start and stop rule in response to tactile stimulation was responsible, at least partly responsible, for the maintenance of processions. But the other thing that you probably noticed from the last videos is, even though in a featureless environment, the caterpillars were able to form processions and aggregate, these processions often lack direction, and the group would often fall apart. So as you can see in this video here, and this is just caterpillars placed on a blank sheet of paper with no other stimuli. They kind of went off in all kinds of different directions, and usually the group would splinter and you would end up with multiple subgroups. So based on this, we hypothesized that there was another factor that was necessary for the maintenance of group cohesion and the direction of processions, and this was the chemical signal that we had previously hypothesized. So we wanted to see how these findings applied to experiments done on individuals and pairs of individuals. So we moved on to, first of all, test whether single caterpillars were able to follow chemical trails. So we created a pheromone extract, a chemical extract of each species, just by placing euthanized individuals in ethanol and using that to swipe the arm of a binary choice maze. So we drew up a maze like this on a piece of piece of paper, and one side of the maze was swiped with pheromone mixture, and the other side was swiped with just solvent. And we tried to see whether each species was able to follow a chemical signature of the same species. And we found that indeed each species preferred arms of the maze that were laced with their own pheromone over controls. And we also found that each species preferred to follow the pheromone trail of the other species over a solvent-based control. So that meant that in both instances, there was a chemical signal that was directing individuals, and the signal was mutually attractive. The pheromones of each species were mutually attractive to each other. So this might have accounted for perhaps how both species got together in the first place. Maybe H. nanas actually stumbled upon the pheromone trail of H. metabus, and that's how they may have joined the colony in the first place. So this supported the hypothesis that chemical signals were involved in directing processions and also maintaining group cohesion, and again, that both species had mutually attractive pheromones. So we wanted to see how this pheromone effect scaled up to a bigger group. So this picture, so if we look at this picture at the bottom right, we created a pheromone template. So the kind of this arm up here was laced with pheromone, and this arm down here was just laced with control. So we wanted to see how a chemical template would affect the behavior of a bigger group. And we found that there was this really nice reaction where they directly followed the pheromone, and they walked all the way to the end, and then they would turn around and come back. So nothing to see there, and they turn around and come back. And then they would loop back and forth doing this. Okay, so the second thing that we wanted to test in pairs of individuals, in just pairs in procession, is do caterpillars consistently follow the start and stop rule, right? Because before, when we did it, it was we hadn't done it in a quantified way. We just saw that it happened most of the time, but this time we wanted to see if this was a statistically significant response. So we allowed pairs of individuals to form processions. So up here we have a picture of one metabus and one nanas individual in procession. And we tried the same manipulation where we held back the one in the back and recorded the response of the one in the front, and then allowed them to rejoin. So we recorded how many times they stopped in response to contact being lost, and started moving off again in response to contact being reinitiated. And indeed, we found that they did, both species consistently followed this start and stop rule. And we wanted to confirm that it was based on tactile stimuli. So we did a second set of experiments where we allowed pairs to form processions again, held back the one in the back, and replaced it with tactile stimulation to the rear of the one in the front. And again, they showed this consistent start and stop response when they were either when stimulated to the rear or when stimulation was removed. So this also confirmed the presence of shared local tactile rules in the maintenance of processions in both species. So far, we've basically found that both species seem to follow a lot of the same behavioral rules in response to tactile and chemical cues. But here's where they actually diverged. So we wanted to figure out, we know that both species are responding to tactile rules and chemical rules in their processions, but we wanted to know if we gave them contradictory tactile and chemical stimuli, which one they would prioritize. So we kind of suspected that, considering the fact that they usually stayed together as a group, maybe they prioritized tactile cues over chemical cues. Otherwise, we might expect them to just zoom off up their pheromone trails and not stick together. So how we tested this was we did the same experiment with pairs of individuals and testing the start and stop rule. But this time we placed them on a pheromone trail. So kind of like the picture in the corner there, but just a straight line of pheromone drawn onto a page. And we placed we put the two individuals on the trail and tried holding back the follower and recorded what the one in front did. And we compared this to what they did in the absence of a pheromone trail. So for H. nanas, what we found was that even when they were on a pheromone trail, they were still equally responsive to to the loss of the tactile stimulation, right? So for H. nanas, when you put a pair on the trail, on a pheromone trail, when you held back the one in the back, the one in front always stopped anyway. So it was the response was essentially the same whether or not it was on the pheromone trail. But the interesting thing was that for Hylesia metabus, there was a very different response. So without pheromone, Hylesia metabus consistently responded to the stimulation from the follower, so stopping when contact was lost and continuing when contact was reinitiated. But when we put Hylesia metabus pairs on a pheromone trail, they didn't really have much time for the follower after that. So in more than half of the cases, when the follower was held back, they just stormed right ahead and they just followed their trail. So from this, we concluded that Hylesia metabus is significantly less responsive to tactile cues in the presence of conflicting chemical cues, and that Hylesia nanas is always responsive to tactile cues. So they prioritize tactile cues over chemical, and in H. metabus is more of a mix. So they're kind of equally responsive to both. And we think that this might have some bearing on the kind of global behavioral patterns in both species, because in wild colonies of Hylesia nanas, we don't tend to have so they do move between resting and feeding sites as a group, but they don't have these long-term home patches in the way that H. metabus have. So H. nanas um, tend to stay in one place for a little while and then move off to a different location. So we think that because of this more nomadic lifestyle of H. nanas, perhaps this is why they're prioritizing tactile cues over chemical, because they have to abandon their old pheromone trails every few days and they need to pay more attention to where their nest mates are more than the trails that they're following. Okay, so I know that was a lot, but basically all of this comes together to suggest that there does seem to be a self-organized mechanism for group coordination between these two species, at least partially shared. And individuals of both species spontaneously form processions and aggregations just through the interactions between individuals. And we can see this even in group sizes of as small as 2. Tactile signals are involved in the maintenance of processions, and chemical signals are also involved in the directing of processions. And we didn't want to bog you guys down too much with this, but we also did a series of experiments that showed that light did indeed mediate the transition to between resting and processing, but it wasn't an instantaneous response. We found that there was a kind of internal clock mechanism that was mediated by light that mediated that transition. And so what don't we know yet? We haven't been able to figure out under what conditions individuals deposit pheromones. So as you probably noticed, the experiments that we did using the pheromones, those were chemical extracts made out of dead caterpillars. So we don't actually know whether caterpillars continuously deposit pheromone or if they only do it when they've been fed or when they're with each other. So we don't know when they actually deposit pheromone, and we don't know how they got to the base of the tree in the first place. So we know that Hylesia metabus females lay their eggs up in the top of the tree. And they around in stuff 4 they walk all the way to the bottom of the tree, and that's how we think they set up this initial pheromone trail, but we don't know how they actually got down there. So there's something else going on, maybe some kind of negative geotaxis. So maybe they just at that age just have an innate tendency to walk down, or maybe they are trying to avoid light. So maybe when they at a certain stage in their life they become very sensitive to light. So those are some of the things we think might be going on, but we don't really know how they got down there in the first place. And perhaps the most kind of glaring question is, what is the deal with these mixed species groups of Hylesia? Are they adaptive? If they are, why don't we know more about them? And if they're not, why haven't they evolved different mechanisms of communication to prevent these kinds of mix-ups? Because these Hylesia species, they use a lot of the same hosts and they have similar geographical ranges. So why is it that they haven't diverged in their communication mechanisms, and is there scope for some kind of parasitic relationship there? Okay, so to sum up, a picture is starting to emerge of a shared self-organized mechanism between these two Hylesia species based on chemical signals, tactile signals, and light. And the overall behavioral differences in these systems, so the more nomadic approach of H. nanas versus the more kind of having a central home site approach of H. metabus, may actually arise from very subtle differences in the hierarchy of the different rules. So the prioritization of tactile rules in Hylesia nanas and the kind of more of a mixed approach in H. metabus. And finally, these kinds of natural history observations can be really useful in helping us understand animal behavior, because a lot of the time when things go wrong it's when we really learn something interesting about the behavior, which is what we think is happening here. And thanks for listening. That's it. If anyone has any questions.